Hello, everyone. My name is Amir Rudic, and I would like to thank you for coming to my talk discussing biocompatible, resilient, and tough nanocellulose-based hydrogels. Now, just as a general overview, hydrogels have been looked at for many different applications, among which is tissue engineering. So in tissue engineering, we're still facing an immense challenge in trying to mimic the behavior of natural biological tissues. These tissues vary in mechanical performance, they vary in shape, and in a lot of different things. Now, for any intended material for these applications, we need to have a way to predict and control the mechanical performance. But we also need to have a material that is able to provide a biocompatible environment for cells to attach, adhere, proliferate, and eventually grow and replace the infected tissue. Of course, ideally, we would also like this material to be able to slowly degrade, be reabsorbed into the body over time as the tissue heals and recovers. We would also like this material to be able to be molded into complex shapes, such as the heart valve you can see in the image below. So, of course, not all our tissues have these perfect, easy shapes. And of course, ideally, we would want our material to be composed of cheap and abundant materials that we could actually use and make this material able to provide it for as many people as we can. So the base material we're using in this project is actually nanoparticles derived from cellulose. So cellulose is the most abundant biopolymer on Earth that can be found in the cell wall of all plants, and is actually what gives them their mechanical robustness. For instance, for trees to really withstand this weight that they need to hold up. Now, cellulose itself can be used as a source material for nanocellulose. So cellulose mass can be actually disintegrated using various chemical or mechanical treatments to yield different nanoparticles. So for instance, if we would take a cellulose mass and expose it to acid hydrolysis, we're able to isolate these small nanometric crystals known as CNC or cellulose nanocrystals, which are very, very strong, thin needle-like rods that in fact have mechanical performance that is similar to Kevlar. On the other hand, if we take the same cellulose mass and expose it to mechanical grinding, we're able to isolate small nanometric fibers known as cellulose nanofibers or CNF, which are still very strong, but they're much looser and enable sort of, again, this fluidity that we still see in plant cells. So of course, these two particles result in different aspect ratios, which in turn affect the viscosity and the homogeneity of a dispersion of these nanomaterials. But at the end of the day, we're still looking at the same basic components. So these two materials are still composed of cellulose, which in turn is composed of very long chains of just glucose units, which with the abundance of hydroxyl groups that are found on them provide us a very versatile chemical backbone to do a lot of different chemistries onto them. So in our specific case, we're actually applying polymer grafting onto these cellulose chains, which is actually carried out through a radical oxidizer in the form of cerium. And in fact, if you look at the top image, well, you can see that by applying a certain form of cerium with mixing it with the cellulose chains, we're able to actually open some of the glucose rings, making them available for polymer grafting. In our case, we're actually grafting an acrylic monomer, acrylamide, which can be seen in the top image. And what we end up with are our cellulose particles onto which we have actual initiation sites of acrylamide. Then, by using the same radicals from oxidation, we're able to slowly form long chains of polyacrylamide, finally ending up with a very homogeneous network of polyacrylamide cross-linked by its usual crosslinker bisacrylamide with CNC and CNF embedded all throughout the matrix, resulting in a very, very strong material that is very different from just pure acrylamide. Now, as I've said, depending on the different nanomaterials embedded, whether it be CNC or CNF, we're actually able to form a whole range of differently behaving hydrogel composite. As you can see in the image above, we're actually able to form a series that, again, depending on whether it was CNC or CNF that was embedded, behave differently. We then tried to perform both compression and tensile mechanical tests on hydrogel samples. And what we saw is that we are actually able to really tune the mechanical performance of the samples depending on the, again, on which nanomaterial we embed into it. So if you look at, for instance, the purple sample, C1F0, where CNC alone was embedded, you can see that we are able to make a relatively soft but stretchable material. On the other hand, when we embed CNF alone in the orange samples, C0F1, we suddenly get a whole shift, so a material that is much less stretchable, but significantly stronger. But then when we embed different ratios of the two materials within the system, we're actually able to benefit from both these behaviors. So if we look, for instance, at the green samples, C3F1, we're actually able to form materials that can both stretch significantly longer, but also are just as hard as embedding CNF alone. Now, as I've said before, this is a material that is actually a hydrogel. So this material is in fact 90% water and just 10% dry material. This material is also highly porous, which again is something very good for cells to really attach and penetrate onto. But we're also able to see that we can actually dehydrate the samples and then rehydrate them again without losing any mechanical performance. This is something that could be very interesting in terms of biomedicine, if you can envision certain structures that can be, formed, then dehydrated and significantly lower their volume, and then rehydrated again within the patient once it's implanted. Then, as I've said, we set out to characterize the biocompatible aspects of the material. So, just like I said before, it doesn't help us only to have a material that performs mechanically well. We also needed to provide an environment that cells can attach and proliferate onto. So in this case, we actually took all the different ratios that we had and compared them to the base material acrylamide alone, AM, here in the yellow samples. And what we saw is that while acrylamide alone can only facilitate very, very small cell growth for a period of 7 days, and the cells are all clumped together and don't really, let's say, like the environment. Once we embed either CNC or CNF, we can see the cells start to spread out and really start to form colonies and start proliferating. Then, once we add a mixture of both CNC and CNF, for instance, in sample C1F3, we can really see the cells liking the environment, really spreading out and sort of covering the entire hydrogel, at least supporting that this material can be biocompatible and can allow cells to grow and proliferate. What we also set out to do is try to image the cellulose within the hydrogel. Here you can see it in cyan. And then we tried to sort of see if there's a correlation between where the cells are and where the cellulose is. And I think you can appreciate in the image above and kind of both images. Now there's a really big correlation between where the cells are and when the cellulose is, further supporting the fact that cellulose itself can grant some biocompatible properties to many initially inert materials. That's it for my presentation. I'd like to thank you for listening up to this point, and I hope to receive questions from you. Thank you.
So, hello everyone. My name is Débora Ferreira and I'm a postdoc at Center of Biological Engineering, University of Minho in Portugal. I'm here today to talk a little bit about the potential of exosomes in triple-negative breast cancer therapy. In 2020, breast cancer has surpassed lung cancer as the most commonly diagnosed one. About 5,076 new cases were diagnosed in Europe, with an estimated 1 in 11 women in the European 27 developing breast cancer before the age of 74. Another alarming fact is that approximately 21% of these cases occur in women under 15 years old. One of its subtypes in particular, the triple-negative breast cancer, already accounts for 15% of these cases. The relevance of this subtype of breast cancer lies in its features. Cancer that tests negative for estrogen, for progesterone, and the protein called HER2 are known as triple-negative. Since they lack these receptors, these triple-negative tumors do not respond well to the targeted therapies. This type of cancer indeed is more prevalent in young people, in individuals of African and Hispanic descent, and also carriers of the mutation of BRCA1 gene. Despite the enormous advantages that we have, we can see in science, conventional chemotherapy is often still the frontline treatment for this type of breast cancer, that is normally administered in a neoadjuvant way to shrink the tumor before the surgery. Depending on the stage of the disease and other factors, radiation therapy and adjuvant chemotherapy may also be administered. Given that these therapies have some undesirable side effects, as we know, as well as limited efficacy, research has been conducted to find new, as well as more effective and selective therapies for breast cancer. So the main goal of my project is to contribute to the advancement of the triple-negative breast cancer research field. through the assessment of the potential of these of exosomes as drug delivery vehicles of RNA interference-based therapeutics, as well as the analysis of relevant signaling pathways involved in cancer proliferation, invasion, migration, and survival. So, regarding exosomes, they are extracellular vesicles, small extracellular vesicles that are naturally released from all cells as part of their ordinary physiology, as well as during acquired abnormalities. They are usually characterized by the expression of specific marker proteins, including the tetraspanines CD63, CD81, and CD9, and they have been exploited as promising delivery vehicles due to their biocompatibility as well as natural role in intracellular communication. They are known by their ability to surpass biological barriers. They have been safely administered to patients with graft versus host disease, and also they have proven innate pro-regeneration and anti-inflammatory features. Based on that, we started by collecting exosomes from a non-cancerous cell line. They're called BJ, the BJ cells, and we obtained exosomes, as we can observe here by these experiments, with approximate size of 110 nanometers, falling within the accepted limits for exosomes, more or less between 30 and 150 nanometers. We also demonstrated that these exosomes possess the typical exosomal markers, as I referred, the CD9, CD63, and CD81. And furthermore, we also showed that these exosomes can be internalized by triple-negative breast cancers, in particular the MDA-MB231. So then, we validated the potential of exosomes as drug delivery vehicles of a molecule, an siRNA molecule called SCI-MEC1, that targets one of the principal nodes of the MAP kinase ERK pathway, the MEC1, as we can observe here. What we found was that these modified exosomes with this SCI-MEC1 indeed led to a cell death, an impact on cell proliferation in 3 different triple-negative breast cancer cell lines, here highlighted, as well as led also to a reduction of their migratory as well as invasive capacity. Additionally, the methodology called CAM, that came from the chorioallantoic membrane of chicken embryos, was employed, which has been used for various biological assays to study the effect of substance on tumor development, tissue vascularization, angiogenesis, among others. So we started did a small window on the eggshell on day 3, then we implanted the MDA-MB231 cells, we did the treatments on day 12, and on day 17 we analyzed the results. What we observed that was when we treated exosomes, as we can observe here, we modified with SCI-MEC1, a clear reduction here on the tumor size, as well as the number of blood vessels was compared in comparison with controls, highlighting the capability of exosomes as carriers of molecules. To complement the above-mentioned quantitative results, immunohistochemical analysis was also accomplished, and as we can observe here, a clear reduction of the proliferation marker, the Ki67, as well as decrease of lectin immunostaining of IXO-MEC1 condition in relation to the untreated condition was observed. These results are in a perfect balance with the regression of the tumor area and the number of blood vessels above mentioned. So as a final remarks, I would like to say that exosomes are a class of extracellular vesicles that are naturally released from all cells. The outstanding exosome roles, including natural material transportation features and also the exceptional biocompatibility, has guided researchers all over the world to exploit their potential application as delivery vehicles of several different molecules. Their intrinsic properties as biostability, minimal immune response risk, multiple cargo loading ability, as well as being amenable to surface modification, make exosomes a novel opportunity as drug delivery vehicles. Importantly, that great efforts have been done to accelerate the transition of these exosome-based drug-delivered nanoplatforms into the clinic. Indeed, although there is still a long path, engineered exosomes already started to enter clinical development, which makes a true milestone for the entire exosomes field. Regarding our work, we showed that the BJ-derived exosomes indeed presented the surface expression of the tetraspanin markers CD63, CD9, and CD81. The exosome-mediated suppression of the central nodes of the central node of the MAP kinase ERK pathway led to a tumor regression, followed by a decrease of angiogenesis in vivo using a CAM model. And to the best of our knowledge, this is the first report showing the feasibility of using a CAM model to validate exosome delivery systems of sRNA molecules. I would like to thank everyone that's helped me and contributed to this amazing journey. Thank you for your attention.
Hello everyone, I'm Shivesh Anand, a postdoctoral researcher at Aarhus University in Denmark. Today I'm going to give a flash talk on the topic nano-engineered biomimetic scaffolds for tympanic membrane regeneration. This work was conducted during my PhD at the Merlin Institute in Maastricht in the Netherlands. First of all, let's understand what's a tympanic membrane. It's a thin tissue lying at the intersection of the outer and the middle ear, and its main function is to capture sound waves from the environment and transform them into mechanical motion. Even though being a thin tissue, the tympanic membrane is a trilaminar structure with outer epithelium layers and an inner lamina propria. The lamina propria is composed of radially and circumferentially aligned collagen fibers, which are responsible for the successful sound conduction by the tympanic membrane. There are many injuries that affect the tympanic membrane. Among them, microbial infections are the most prevalent one. Here is an example of a microbial infection which affects the middle ear. In this infection, the area behind the tympanic membrane is filled up with a fluid, which is then discharged through the tympanic membrane, thereby creating a hole in there. During my PhD, the focus of the project was to develop different biofabrication strategies for the treatment of these tympanic membrane perforations. We looked at 4 different routes for treating the tympanic membrane for perforations. The first approach was to understand the geometry that is needed for biofabricating these tympanic membrane constructs. The next was to identify the right biomaterials. The third aspect of biofabricating tympanic membrane structures was to include drug delivery capabilities in them. And the final and the most important one was to use external stimuli to recreate the external extracellular matrix of the native tissue. As I mentioned, the first aspect was understanding what is the right geometry for creating these tympanic membrane scaffolds. In this study, we investigated different three-dimensional models and performed theoretical simulations on them. Based on these theoretical simulations, we biofabricated the tympanic membrane constructs using a combination of electrospinning with additive manufacturing. Finally, on the biofabricated constructs, we performed mechanical, acoustical, and biological characterizations. For the geometry, we used the radial and circumferential alignment of the tympanic membrane as our reference. We selected 4 simplistic geometries, where the first was a control with no pattern in them. Case 1 only had circumferentially aligned fibers, case 2 only had radially aligned fibers, and case 3 was a combination of case 1 and case 2. Overall, in the mechanical properties, we saw that as we moved from control to case 3, there was an increment in the Young's modulus, finally resembling what was measured or reported for the native tympanic membrane. In terms of the acoustical properties, we didn't see much of a difference between the different cases that we investigated, but all these tympanic membrane scaffolds remained very similar to the properties that were measured for the native tissue. Finally, the biological characterizations were performed using human mesenchymal stromal cells, where we saw by day 7, all the scaffolds were completely covered by the human mesenchymal stromal cells. And by day 14, these cells started to produce their extracellular collagen, which was aligned along the radial and circumferentially fabricated tympanic membrane constructs. Based on these studies, we concluded that geometry plays an important role within the tympanic membrane scaffolds. The next part was to expand the biomaterials that have been used for tympanic membrane reconstruction. We used PEOT-PBT copolymer as the base material previously. We wanted to expand this material by including a carbohydrate-based nanofiller in the form of ketene nanofibrils. The use of ketene nanofibrils provides 2 different advantages. The first, in terms of mechanical reinforcement, which was confirmed using both indentation and tensile measurements. In both the cases, we saw that the inclusion of ketene nanofibrils drastically improved the mechanical properties of the tympanic membrane scaffolds. The other advantage of using ketene nanofibrils is in terms of its immunomodulatory properties. We saw that the inclusion of ketene nanofibrils in the tympanic membrane scaffolds downregulated all the pro-inflammatory cytokines which were tested in our study. The third aspect of tympanic membrane reconstruction is creating tympanic membrane scaffolds with drug delivery capabilities. At the moment, for the treatment of microbial infections which affect the tympanic membrane, there are 2 different routes. First one being systemic, where the patient takes the antibiotics orally. The other approach is the topical approach, where the patients are exposed to eardrops for treating these microbial infections. However, in both these cases, the patient is exposed to very high dosage of the drug, which might have different side effects. Therefore, the strategy implemented in this part of the project was to combine drug delivery with tissue engineering, which could then result in creation of tympanic membrane patches that could provide precise and targeted delivery of the antibiotics. In this case, we investigated 4 different modalities of electrospinning. The first modality was the control, where we were just electrospinning the PEOT-PBT copolymer. Mode 2 was that of blend electrospinning, where the antibiotic, which was ciprofloxacin in this case, was blended into the polymer mix. Mode 3 was that of simultaneous co-electrospinning, co-electrospraying system, where we used drug-loaded PLGA nanoparticles being electrosprayed from the other end. And mode 4 was a combination of mode 2 and mode 3. In terms of the drug release profile, we saw that with mode 2, we could achieve a sustained release of the drug up to day 7. However, with mode 3 and mode 4, which included that of drug-loaded, electrospraying drug-loaded nanoparticles, we obtained a more burst release. In terms of the antimicrobial validation, we saw that all these different modes that we investigated were capable of inhibiting the growth of bacteria, both gram-positive and gram-negative. And this was accompanied with biological characterizations using human mesenchymal stromal cells, where we saw that there was no detrimental effect on their growth and proliferation. The final aspect of the tympanic membrane reconstruction project was to investigate external stimuli which can be used to promote the differentiation of human mesenchymal stem cells or stromal cells towards the tympanic membrane lineage. In this case, we used 3 different stimuli in the form of molecular, hierarchical, and acoustical stimulation. The molecular stimulation was provided using bioactive agents in the medium. The hierarchical stimulation was provided using hybrid scaffolds, which were fabricated by combining additive manufacturing with electrospinning. And the acoustical stimulation was provided by stimulating the tympanic membrane scaffolds cultured with the cells with sound waves in the audible range. For the acoustical stimulation, we fabricated or we developed an in-house bioreactor setup, which we recently patented. Using this bioreactor setup, we could stimulate the tympanic membrane scaffolds at different frequency range. Here is an example where we stimulated the scaffolds in the range of 1 Hz to 4,000 Hz, and we observed that at certain frequencies, the tympanic membrane vibrated much more drastically as compared to the other frequencies. These frequencies are known as resonance frequencies. This was observed both for experimental measurements as well as theoretical simulations. Finally, we applied an integrated stimulation by combining the acoustical stimulation with hierarchical and molecular stimulation. Overall, we observed that we could downregulate the expression of collagen 1 and upregulate the expression of collagen 2 upon the application of different stimulation routes. This is desirable for the extracellular matrix towards the tympanic membrane regeneration. Finally, to conclude, I would like to summarize that geometry plays a critical role in fabricating biomimetic tympanic membrane scaffolds. Moreover, chitin nanofibrils could be used to mechanically reinforce and provide immunomodulatory properties to these tympanic membrane scaffolds. In addition, we saw that the tunable drug release properties could be included in the tympanic membrane scaffolds, which could be used for the treatment of microbially induced perforations. And finally, we observed that advanced cell culture systems can be developed to stimulate the stem cells differentiation towards tympanic membrane regeneration. I would like to conclude this flash talk by thanking my supervisors, Carlos and Lorenzo, along with all my team members at the Merlin Institute, along with my collaborators in Italy and Germany. Thank you for your attention. I would be happy to answer any questions if there are. Thanks.
Hello everyone, my name is Andrea Cabero and I will present my project called Optimization of Chaotic Bioprinting to produce reinforced multi-channel hydrogel fibers for the maturation of skeletal muscle tissue under agitation. Chaotic bioprinting emerges as a novel technique to print multiple materials simultaneously in a single step. It helps us to produce prevascularized hydrogel fibers with void microchannels that resemble the natural vascularization of tissues. However, this type of fibers do not have good mechanical stability and degrade after some days of static culturing. For this reason, the main objective of the project is to optimize chaotic printing to produce reinforced hydrogel hollow fibers that can sustain skeletal muscle tissue under agitation. We used a Kennex static mixer of 8 inlets and 2 mixing elements as our print head. The mixing elements induce the chaotic flows to compartmentalize all the materials into just one single structure. And when the printing was done, we placed the hydrogel fibers in a rocking bioreactor. First of all, we optimized the printing system. We used a printing rate of 1 milliliter per minute, and we have 3 biomaterials. Alginate as our structural material for improving the mechanical properties of the structure, hydroxyethyl cellulose, or also known as HEC, as a sacrificial ink, to fabricate void microchannels that resemble the vascularization, and a combination of Gelma and alginate as a bioink for cell adhesion. So we had 8 inlets and 3 materials, so we could change the proportion of all of these 3 materials, and we compared the conceptual and experimental cross-sections, in which we saw that we could estimate the distribution, shape, and proportion of each layer inside the hydrogel fiber. So we could, in this sense, tune the composition and the microarchitecture of our structure for the application, any application we want to give it. And the proportion of each ink was similar between the conceptual and experimental cross-sections. And then we optimized the proportion of alginate because it was our structural material. We did tensile assays and we obtained the stress-strain curve by changing, we used samples that change, in which we changed the proportion of alginate, and we used one control sample made of just alginate as a control, and we concluded that the optimal proportion was to use 3 out of 8 layers of alginate inside the fiber, because it gave us a sweet spot between mechanical stability but also including as much void microchannels that we wanted, because the void microchannels improve the mass transfer of nutrients for the cells, and also to don't miss out the bioink layers, because in these layers cells will be adhered. And the elastic modulus of this optimal fiber was 12.8 kilopascal. So then we did the cell culturing of mouse myoblasts, and we compared reinforced solid fibers. Reinforced means that it had structural material layers, and reinforced hollow fibers with the void microchannels. In the solid fibers, we observed in the actin Dapris staining at day 14 in static culturing conditions that we have cell elongation and spreading, but just in the surface of the fiber, because the thickness of this layer with all the cells was not more than 60 micrometer, and this happened because we didn't have void channels and that mass transfer of nutrients was not good. However, and luckily for us, when we used reinforced hollow fibers, we could observe that the cells start to spread at least in a volumetric distribution of 400 micrometers, which is good because it is an indicator that we had a better mass transfer of nutrients. Finally, we compared the cell viability of static versus agitation culturing conditions. So when we just assess the cell viability of fibers under agitation for day 1 and day 7, we saw that for both solid and hollow reinforced fibers, we have good cell viability because most of the cells are staining green, which is meaning that they are alive. And then we compare static versus agitation, and we saw that for both solid and hollow fibers, the agitation improved the cell viability at day 7. Thank you so much.